Okay, so next lecture, the probe effect. There are two parts to this lecture. I'm only going to get through the first, I think, the way I'm going. Okay, so here is a plot of correlations between, must be like 200 to 500 arrays. So these are from the Geo repository. There's maybe one that isn't, but these are all public data. They've all been downloaded, and so I have all the data, and I can compute the correlation between each pair of array. And I'm plotting them as color, where the dark blue is 1, and light blue is 0.5, white is 0, red, there's no red, but red is negative. So these are several experiments, and they include all kinds of tissues. So the first thing that we see that kind of calls out at us is that everything is blue. There's no white, there's no red. That means every single tissue is correlated with other tissues. And that doesn't make that much sense biologically. There's high correlation in most cases. So it turns out that this is an artifact of something that I'm going to describe today, which is the probe effect. All right, so let's take a closer look at what this means. So here are two genes. Now, instead of all this correlation between all samples, I'm actually taking it down to one gene, and that's already two genes. There are the names of the two genes, and down there is a scale of log2 intensity, and now there's a little arrow. That arrow is pointing at the value that I observed in a specific sample. They're both 8. That's what I'm showing you here. So if you see that they're both 8, and I ask you, is this gene, which of these two genes has higher expression in this sample, you would say they're the same, right? That's what you hope, given that that's what we're trying to measure. But I think that answer is wrong. I think that gene number 1 is much higher expressed than gene number 2, even though both of them reported an 8 in the log2 scale. So why do I think that? All right, so what I added to the picture is a histogram of all the values from all the samples that I showed in the previous picture. In the previous picture, there were about 200, 500 samples, right? So now I have, for each gene, I can show you all those values. So in the first one, most of the values are down here, and there's a few out here. In this one, most of the values are down here, and some of them are up here. So biologists in the room maybe can tell me what they think is driving these two humps where most of the data is. What do you think that represents, given what we know about gene expression and specificity to tissues? So would you say that these genes are expressed or not expressed? You think they're expressed? So not expressed. So you're saying no. So I think they're not expressed. So the majority of samples, a gene will be unexpressed in most tissue types. Is I think something that people who know about gene expression kind of agree on. You know, if you look at a specific tissue, 10 to 20% of genes are turned on. So if you look across many tissues, then the chances of a specific gene being on all the time is not that high. But there's another reason why I know this is off, is because included in there are background controls, and they're also falling in there. So of course I didn't tell you that, so you couldn't use that information, but I'm pretty convinced that this is actually not expressed. So remember, this is one gene. That gene is not expressed in most samples. It's expressed in these samples. And here, I would say that these guys are not expressed. All these little ticks, there's a lot of ticks there that you can't see, are not expressed, and then these guys are expressed. So this difference in where the background level is. So notice that the background level, if you agree with me that that is the non-expressed aspect of it, the background level for this gene is at 4. The background level for this gene is about at 7. So it's 8 times bigger. Background is higher for this gene than that. And we saw a little bit of that in the background correction talk, where we saw that a specific two probes for the same gene could have had a very wide range of background levels. That variability passes on to the gene level, because it's only 11 probes per gene. So the probe effect survives the averaging that occurs at the gene level. So that's my interpretation of this plot. This gene is not expressed in this sample. This gene is expressed, despite the fact that they give you the same value. That's what I mean by the probe effect. The probe effect makes the values that come out of gene expression experiments not interpretable on their own, in the absolute. It's a very important thing to know, because mistakes can be made from this. Because if you can't compare the values of two genes just straight, because you will run into this problem. And the same is true for anything else you're measuring. Whenever you measure something with a microarray, you will get these effects that make it hard to interpret. Another way to think about this is to say that the units of this measurement, of the measurement for this gene, is different than the units for this. So this could be Celsius and this could be Fahrenheit, for example. Right? So you don't know what that unit is. So if you don't know what the unit is, you know what the value is. So the way we can actually put a context to those values is to look at more than one sample. This is what I'm doing here. All right, so here's one way to fix this. If you think that every gene is measured in a different unit, then we can get rid of that unit by standardizing each gene. So what we mean by standardization in statistics is we take the row, we have a row of values for a given gene. We take the mean or the median. I think in this case I took the median. You subtract the median, then you compute the standard deviation or the MAD, and you divide by that value. And now you get numbers that are centered at 0 and have a standard deviation of 1. So there's no units. So every gene is unitless and should be interpreted relatively the same way. If we think most genes are off most of the time, 0 will mean not expressed. And something like 3 or 4 or 5 will mean expressed. So once I do that, once I center the genes, once I standardize the genes and I recompute this correlation matrix that I showed you earlier, now we see a lot more white. Right? So now we see that many tissues, when you compare to each other, actually have no correlation, which makes more sense. Now, we see these blocks. You see the blocks? What do you think those blocks are? So every little line here is a different sample, and every column is a different sample. Each pixel is the correlation. The color of the pixel represents the correlation between those two samples. So these, for those that don't know, these are experiment IDs. So all these samples come from this experiment. So this is saying that the correlation within that study is very high. And that makes sense because most of the time a study focuses on a tissue. So that's probably a tissue. The same tissue is being measured multiple times, so of course there's high correlation. So now this is looking much more like what we expect. But we have to remove the batch effect before we can see something like this. So here's that same picture I showed you earlier. This is kind of confirming that there is, I think, that they are off. Here's the histogram that I showed you earlier, and here's for gene 1 and gene 2. I'm adding, this was in the previous picture, but I didn't explain it. The ticks, see these ticks? The orange ones are called absent by the manufacturer. The green ones are called present. That's their their expressed, not expressed call. And M is for marginal, so they're not sure. So here they're calling all these absent. But then there's a lot of other ones that have the same value that are called present. It's kind of strange. And over here, all of them are called absent. I don't trust these very much. And I think this is what I'm showing with this picture. Now, in the next plot, I'm showing box plots where I have aggregated the values shown here by tissue. So every box plot is showing me the tissue, all the values for each tissue. So there's what, like 20 to 30 tissues here? So you can see now that all these tissues, it looks like that gene is off in those tissues. And then there's 2 tissues, 1 and 2, for which that gene is on. And then here the same thing. It looks like these are off, and these 2 tissues, that gene is on. So it kind of gives more credence to the statement that this hump is the off hump, and these guys are the on values. Right, and the title here, barcode, that's because we use this view of the data to define barcodes for gene expression, where you get a 1 or a 0 based on this data, and it is useful if you're interested in knowing if a gene is expressed or not, as opposed to finding differentially expressed genes. My view is that the only way to do that is to look at data in the context of all experiments. If you just have one array, I don't think you can do that. All right. So there's the picture again after centering. Oh, and another little thing I wanted to point out here is that notice that if these guys are all, if it's true that these values come from a gene that's off, that's not expressed, notice that there are differences across tissues. So for example, take this tissue and this tissue. They're little tiny box plots. So say compare, actually we can just stay here. Compare this guy to that guy. So this is a box plot, so there's more than 3 values there, and there's more than 3 values there. So if I do a test on these 2, if I just do a differential expression, what do you think the p-value is going to be? Is it going to be significant? Yeah. It is, for sure, because they're completely separated. So if I do a differential expression analysis of those 2 tissues, that gene will be called differentially expressed in these 2 tissues. But I'm saying they're both unexpressed. So that there's a disagreement there. So which one is it? Are they differentially expressed, or are they both off? I think they're both off, and I think that the difference we see is batch effect. So you can really, that sort of changes you see, that wave that you see across samples, I think that's batch effect. And that's a problem that we will be talking about for an entire lecture, and how to fix that. Which outlier? You mean this guy? Well, I think that's part of this one. So it's a gene that's on, so probably more variable. It's on at different levels. But I mean, I think in general we can say that this tissue, for this tissue, this gene is on. Oh, where are these guys? I think now now that you asked me that question, I believe that what I'm plotting here are the medians. Is it possible? No. So that guy, no, maybe not. I think that guy is maybe this guy. And maybe this box plot is based on just one number. That's possible. Yeah, that's possible. Actually, yeah, so I think these are the individual values. So I would say that these guys are these guys, and this tissue actually may only have one. It's possible it only has one value in it. Okay, so So yeah, so by the way, with the barcode, if you just turn things into zeros and ones, and you recompute the correlation, and you see even more white, which is nice, right? So that means that there's blue when it's the same tissue, and then not that much blue when they're different tissues, which is what we like to see. Okay, so now a real downstream consequence of this batch effect. There was a paper published. Sorry. Whoa. That was a Microsoft move there. Okay, so there was a paper published, I can get you the reference, that claimed they took a bunch of tissues from mice and humans, and they did a clustering procedure on them, and they found that the tissues from mice were more like the actually it's this one. This is the wrong way. I'm sorry. So it should be it's the other way around. So look at this picture first. So this is, if you take gene expression from a mouse chip and a human chip, and you find genes that are homologous, and you match them, and you make a big matrix of gene expression. Right? So you find the genes that match, you make a big matrix, so now you have different tissues from humans and different tissues from mice, the same tissues, and you run it through a clustering algorithm. You get this clustering. So notice what's happening. All the human tissues are here, and all the mouse tissues are over there. They published a paper claiming that this is an interesting biological finding, that the human kidney is more like a human brain than a mouse kidney. That was the claim. Now, those of you who have actually opened a mouse and seen their kidney, you know it's pretty similar to the human one. It makes no sense biologically. So what do you think is happening there? This is a tricky one. Why is that happening? Why are all the human samples clustering with the human samples and the other ones with the other? Yeah, so there's two arrays, right? So the gene for the human array is represented by a different set of probes than the same gene for the mouse array. So they have different probe effects. So what this is saying is that the probe effect is stronger than the biological effect. It wins. Now, once you apply the barcode approach I just explained, where you take each gene and you make it 0, 1 based on the context of the entire chip. So these two, I'm sorry, but these two are interchanged. It should be the other way around. Once you do that, look at what happens. You do the barcode, and then you do the same thing. You cluster, and now biology makes sense again. Liver with liver, kidney with kidney, lung with lung, spleen, no mistakes. So yeah, so that paper is published somewhere. Somebody wrote a letter. It wasn't me. Okay. I'm glad I didn't have to write that one, because all you get is hate. But yeah, somebody wrote the letter saying all you're seeing is a probe effect. All right. Okay, so we have 20 minutes, so I'm going to actually start the next lecture. Oh no, this is not the next lecture. This is the same one. Okay, so on SNP, we actually saw this already when we did clustering. You can see, I just want to show this so that you can see the probe effect applied to something other than expression. On a SNP chip, we see the probe effect again. Here's data from 3 genotypes. There's 270 people. This is one SNP, and we're showing the A and the B intensities. So we can see the AAs, the ABs, and the BBs. And these circles are showing us where these things land. But as soon as we go to another SNP, they move somewhere else. That's the probe effect again. The probes are behaving differently depending mostly on their sequence. So one of the things that affects this one is overall has higher intensities than the other one. You go to another probe, this one has lower intensities. than the other one. So it comes way down here, and then here's another one. And so every probe has a different effect. And if you don't know that, then you won't know that things like this are going to happen.